Welcome. So today we're going to be covering our first of, four lectures about evolution or evolution in some capacity. So today we're going to be covering the evolution of populations and have this awesome population of lions looking extra cute as lions tend to do. So the underlying principles of evolution stem from genetic variation. And what do I mean by genetic variation? Well, we think about genetic variation as simply differences is in the alleles of genes found within individuals of a population. Now, just to refresh your memory, an allele is just a different variant of a gene. And when we think about any given population, whether it's the human population, where there's those cute lions I showed you in the previous picture, there is a considerable amount of variability in genes, in the alleles, and in the traits found in any given population. If we were in person for this, I would have you look around the room, just assess the wide diversity of traits found in your classmates, whether it's hair color, eye color, height, things like that. Now, all this variability is what our friend evolution really acts upon. And evolution, at its most simple, describes how an entity, and this entity is not an individual, but it is in fact a population or a species changing through time. And when we think about how this came to be, it does get traced back to Charles Darwin, and he came up with this idea back in the 1850s called descent with modification. But evolution is the uniting principle for biology. It doesn't really matter what sect of biology you go into, evolution is the knot that ties them all together in some way, shape, or form. So these four classes about evolution are particularly important for those that have any interest in science as a whole. Now, Charles Darwin has a really great quote about evolution, and he says, through time, species accumulate differences. As a result, descendants differ from other ancestors. In this way, new species arise from existing ones. And I think that really encapsulates what evolution is in a nutshell. Change through time, these changes accrue through time, and it allows you to be different from your ancestors. And you might not really know that, but you are different than your ancestors, particularly ones that were, say, a few thousand years ago. Now, evolution as a process leads to change. Now, evolution itself is not directed, it's not like trying to make things better, but evolution does elicit change, and it does change by acting upon natural variability within a population. Now, Darwin himself was not the first to propose the theory of evolution. However, Darwin did propose that natural selection as the mechanism of evolution. And we know this to be true nowadays. But what natural selection really gets at is the environment selecting for traits that are beneficial in that environment. So you can imagine if you live in a desert and you are a really sweaty person. Well, that might be a beneficial trait to have in the desert because sweating allows you to stay cooler. And if the desert's really hot, sweating a lot allows you to stay cooler in the desert. Now, natural selection directly can lead to changes in allele frequencies or the frequency of an allele of a gene from a generation to a generation. So Darwin was thinking about traits when he was thinking about evolution, but we think about evolution now as not just traits, but we also think about it as changing the frequency of genes and the alleles of genes. Now, when we think about genetics, when we think about evolution, we think about population genetics coming together to think how genetics and evolution are intertwined with one another. And we're simply thinking about studying the properties of genes in a given population and evolution resulting in a net change, either positively or negatively, in the genetic composition of a population. Now, natural populations, as we mentioned, do contain substantial genetic variation, and that's a general rule. It's not true for every population. For instance, like the endangered rhinos of Africa have very, very low genetic variation, but as a general rule, a healthy population has substantial genetic variability. And this genetic variability is really what is required for evolution as a mechanism to occur. And we could just visualize this, right? So normally if we were in a class together, I would have you look around the room and see how diverse your classmates are. But we could see that here, right? We could see this just group of people kind of looks like they're at a concert or something like that. But you can see how variable the people are: hair color, skin color, height, right? All these different things, hyper variable. And then we can look at something, more uniform, right? We have this field of flowers where there's, only really a few different types of flower variants, but there is still genetic diversity, genetic variability found in this population. Now, how do we measure genetic variation? Well, you can kind of look at, a level such as this and say, wow, there's a lot of traits, thus there must be a lot of genetic variability. And, by and large, you would be correct. But we do know that we measure genetic variability now by just simply looking at the genome, whether it's the genome of a plant or whether it's the genome of a given human individual. And we can look at some just very basic examples. So some really easy to understand variability in the genetic code that leads to variation in terms of traits is going to be human blood groups. It's a pretty straight one, right? The ABO blood group with with the plus and minus, right? Very, very small and easy things to understand in terms of genetic variation. Now, we can also look at what we call SNPs, or single nucleotide polymorphisms. They're also called SNPs. It just depends on who you are. I'll probably use to call them SNPs because I think that's a fun word to say. And these are used to assess patterns in over 300 species, including humans. So for those of you that have ever gotten an Ancestry.com or one of those, oh, we're going to predict that what your ancestry is based upon your DNA, they use SNPs. And all a SNP is just a single base pair change. That's all it really is. But ultimately, the best way to assess genetic variability in a given population is to sequence the whole genome, right? Sequence every piece of DNA inside of a cell and look at the variability from individual to individual. So, and this last one, it sounds, might sound a little expensive or far-fetched, but the cost of sequencing a genome now is less than $1,000. So it's really not that expensive now, which is a net good thing for genetic variability, but also a net good thing for understanding human diseases and things like that. So back to Darwin. Now, you guys might remember from Biology 1, a gentleman named Gregor Mendel. Now, Gregor Mendel is considered to be the father of genetics, and he studied peas, right, to understand how traits varied in peas through generation to generation. And the cool thing about Mendel and Charles Darwin is that they were contemporaries. But the interesting thing is that they never met, they never talked, they never exchanged data, they never even exchanged a letter. They were basically unknown to one another. And Mendel's work as a whole was also relatively unknown. Charles Darwin's work got a bit more traction, but Mendel's work was kind of, brushed aside. No one really thought about Mendel's work. And when Darwin was doing all of his things, thinking about genetic variability, he really had no idea about the underlying mechanism of evolution. He saw evolution happening, he saw the patterns of evolution, but he had no way to understand, well, what the heck was going on? How was evolution actually occurring? How are traits being passed on from the next generation? How was there so much variability in traits? And so genetic variability and evolution, as I sort of hinted at, are really tied with one another. And so it's kind of an interesting thing. You could imagine how different a state this field of genetics or evolution would be in if actually Mendel and Darwin actually spoke to one another and exchanged ideas and tried to build their theories together. But, unfortunately, that just wasn't the case back in the 1850s and 60s. Now, at the time, scientists thought selection should favor what we would call an optimal form, that there is a best type of form for any given individual or any given trait, and that that selection itself, whether it's human or natural, should eliminate any variability. But we know that's not the case, right? We know that there's a huge amount of diversity in the population. If there was a best form, nature has not found it yet. We'd also proposed that there was this idea of blending inheritance. And this was something that was extremely widely accepted, that offspring were expected to be some sort of blend between a relative, um, a blend between parents, right? So, think about your two parents, think about if you were an exact copy of both of them smushed together. And that's what blending really is. There's also another idea, its name escapes the top of my brain right now, but it's basically you inherited your traits directly from your father, so that the sperm carried all the traits. So there was ideas here, but after Darwin, there's many other people who thought about evolution. There was a lot of people who thought about how things changed over time. And this led to arguably one of the most well-known principles in all of evolutionary biology, which is what we call the Hardy-Weinberg principle, abbreviated always as WHW equilibrium. Now, Hardy-Weinberg predicts and links together this genotype, so underlying genetics, to the phenotype, the overlying physical characteristics, with five distinct um, properties that populations have to have for the proportions of those traits to not change. So let me say that one more time in a slightly different way. Hardy-Weinberg said proportions of genotypes do not change in a population as long as these five conditions occur. So no mutation, so no underlying changes in the DNA, no genes are transferred to or from other sources, so no immigration or no migration, so no coming in and out of the population. There's always random mating, so there's no like guarantee some animal would mate with this animal. The population size should be very large and no selection occurs, right? So no environment selecting. And that's, these are kind of some lofty goals, right? They make sense intuitively that allele frequencies or genotype frequencies would not change, but these things we know are natural, right? We do know mutation is natural. We do know immigration and migration exists. We do know that mating is not random. And we do know the populations can be big, but not always very large. And we do know that selection by the environment in particular, whether or not it's, happening a lot or a little, is always occurring. So that's the Hardy-Weinberg principle. Now, we can express Hardy-Weinberg as an equation, and we can use it to calculate allele frequencies. Now, what we look at is frequency of the first allele, which we designate as lowercase p, and the second allele, which we designate as lowercase q, is p plus q equals 1. Now, 1 simply refers to 100%, right? So if you think about, 100 as like as a decimal, right, that would just be 1. So we could think about this. P equals big B for black coat color, and so a black cat would be big B, or big B, little b. So that should, refresh your memory from some Mendelian genetics. And then Q is equal to little b for white coat color, and white cats themselves are BB, i.e. little b. Now, remember, this is looking at allele frequencies, not phenotypic frequencies like coat like black or white, but it's looking at allele frequency. So we can express this system, this very simple system, as p squared, because we know we have pp plus, I'm sorry, bb, sorry, not pp, right? P squared, or bb, plus 2pq, so 2 times b, which would be p, and then little b, which would be q, and then that's plus q squared, which gives us bb, or little b. And that all is equal to 1. So we can derive any genetic set by simply denoting the dominant allele as p, the recessive allele as q, and expressing this equation, right? Because if you have two distinct alleles, there's only 3 possibilities, right? If you have 2 distinct alleles here, there's only dominant, recessive, and recessive, right? There's no other unique combinations here. Now, this equation would change if you had more alleles or if you had a more complicated system, right? But in terms of this very basic system, which we're going to be thinking about, we associate with P with the dominant allele, Q with the recessive allele, and we can express their frequencies with this mathematical equation called Hardy-Weinberg equilibrium. So let's look at this. Let's do a quick little problem. So we have some phenotypes: 2 black cats, 1 homozygous dominant, 1 heterozygous, and 1 homozygous recessive. The frequency of its genotype in the population is 36% for our dominant, 48% for our heterozygote, and 16% for our recessive. So the frequency of gametes in the population, so things that gave rise to these individuals, is 36%, 24%, which would give us the frequency of B, and 24% plus 16%, which gives us the frequency for lowercase b. Together, you'll notice that 40 plus 60 gives you to 1, or 100%. So that's how we do our math. Now, just to highlight how I got these 24%, right? The frequency of this gamete is 48%, but half of it is a big B, half of it is a little b. So you divide that 48% in half, because half of it is big B, half of it is in fact little b. So let's look at another example of how we would calculate this. We have a sperm and an egg, two heterozygotes, big B, little b, big B, little b. The frequency is 60-40, 60-40. Now the probability, so this is in the parental populations, right? Big B, little b, 60-40, big B, little b, 60-40. Now, which is 24, so 0.24, fun stuff. Same, oops, sorry about that. Same thing in this quadrant. And then finally, this quadrant, we have 0.4 times 0.4, giving us 0.16 for our q squared, or little b, little b. So this is how we get this equation, right? P squared, because it's big B times big B, little b times big B, which gives us PQ, but we have two PQs here. And then we finally have little b, which is point Q times Q, or Q squared. So that's how we derive this Hardy-Weinberg equation. It's pretty, I don't know. I always, when I learned this way back in the day, I always thought it was kind of abstract until I thought of it in this fashion with the Punnett square. So whatever helps you remember and disentangle how we get this equation is by far going to be the best way to do it. Now, if all 5 assumptions for Hardy-Weinberg equilibrium are true, all allele and genotype frequencies do not change from one generation to the next. Now, as I mentioned, most populations simply cannot meet all 5 assumptions of Hardy-Weinberg. They just can't, right? All the things that we talked about, those 5 assumptions, right? They're all happening pretty much all the time, everywhere. However, the primary use of this equation is to determine whether an evolutionary process is operating in a population and potentially understand what those are. So we think of Hardy-Weinberg not as what could be happening naturally, but we think about Hardy-Weinberg, what's happening in the absence of evolutionary change. So we can compare what's happening in a natural population that is evolving, that's not under Hardy-Weinberg equilibrium, or Hardy-Weinberg equilibrium, and compare it to what's happening in a population that is not undergoing evolutionary change, so we can understand that mechanism of evolutionary change. So Hardy-Weinberg is kind of nice. Now, what makes populations differ under Hardy-Weinberg equilibrium? Well, there's lots of different reasons, but the most common ones we see include natural selection, might favoring homozygotes over heterozygotes. It's pretty common. Individuals may choose to mate with genetically similar individuals. So there is no random mating. That's pretty common, right? People in, say from Scandinavia tend to mate with people from Scandinavia, right? And maybe people from New York tend to have children with people from New York, right? That's non-random mating. There's always an influx of individuals from other populations, so immigration and migration are usually constantly happening as a general rule. And mutation is always occurring. And whether it's very small mutations or very large mutations, mutation is kind of always this background process. And this is just a handy little figure / table looking at the agents of evolutionary change, whether it's a mutation, which is the ultimate source of variation, right? We have all these variability in traits because mutation changes DNA. Without mutation, nothing good happens, right? And we have all these traits, and we'll discuss these to varying ability throughout the rest of the semester. But just keeping these ones in mind: mutation, gene flow, non-random mating, genetic drift, and selection. So let's talk about these a little bit more in depth, but we'll again, these are themes we're going to discuss throughout the entire course. Now, mutation rates are typically low. Now, they're low depending on the population. It's kind of hard to quantify mutation rates always being low, but they're sometimes fast, they're sometimes slow. They can be incredibly fast terms of changing allele frequencies, it's not overall that important to changing allele frequencies, but it is important for changing DNA. And it does, again, makes evolution possible. Now, that's one. Second up is gene flow. And this is the movement of alleles from one population to another, where animals or individuals physically moves into a new population. Now, this would be the functional equivalent of someone moving from the United States to, say, India, right? Bringing the genes of people that live in the United States to India, which is an important process, right? Certainly for humans, but it's also an important process for natural populations, right? You can imagine if you have a population of birds on one side of the Rocky Mountains, if some of those birds migrate over the Rocky Mountains, they're bringing new genes to the other side of the Rocky Mountains. Now, the drifting of gametes or immature stages of plants and animals is a pretty common way this happens. So, like a current carrying crab gametes down a current in the ocean, or a plant's pollen or seeds wafting over a mountain or something like that. But it does allow mating of individuals between populations that are not normally intersecting with one another. Now, non-random mating has two categories underneath it, which we call assortative mating and disassortative mating. Assortative mating tells us that phenotypically similar individuals will mate, and this increases the proportion of homozygous individuals in the population, which is in contrast to disassortative mating, which is phenotypically different individuals mate, which produces excessive heterozygote. So it just depends. Some populations prefer one, some populations prefer the other. It just really depends. Genetic drift, which is an important concept to think about, which typically occurs in small populations, but it can occur in medium to large populations as well. Allelic frequencies may change by simply chance alone, which is which is a fun thing to try to quantify, but it is a thing, right? Chance probability plays a role in pretty much everything. But remember, to satisfy Hardy-Weinberg equilibrium, populations have to be large. Now, the magnitude of genetic drift is inversely related to population size. So genetic drift is a much bigger deal in smaller populations than it is in, say, medium-sized populations. Genetic drift itself can simply lead to the loss of alleles in very isolated populations, and uncommon alleles are also more vulnerable. And this can lead us to two distinct categories of genetic drift, which we call the founder effect and the bottleneck effect. Now, the founder effect. Usually I put these things in alphabetical order, but I went founder first, then bottleneck. Founder effect tells us that one or few individuals simply disperse and become the founders of a new population. That's kind of nice, right? Think about if you have a population of, say, mice drifting on a piece of driftwood in the ocean and they land on an uninhabited island, right? That's a founding population. That's the founder effect. Now, you can imagine in that scenario where mice are floating on a wood plank on the ocean, that's a very small subset of the original population in terms of genetic variability. So in the founder effect, alleles are lost and causes other alleles to change in frequency. And so it's pretty, it's not uncommon for this to happen. Now, we know that it happens on islands. We also know this can happen with self-pollinating plants, so plants that do what we call selfing, so fertilizing their own eggs. We've also seen this happen in the Amish population. So the Amish populations have, migrated to remote areas or distinct areas, and they kind of stay there, limiting their gene flow in and out. So lots of ways that can happen. Now, the bottleneck effect, as its name kind of implies, is it's like pouring liquid out of a bottle, right? If you're trying to rapidly pour liquid out of a bottle, the way a bottle is designed, it limits how much liquid can flow out at a given time. And that's really what the bottleneck is getting at. It's a drastic reduction in population size due to natural forces. And these can be drought, disease, hurricanes, things like that. So there's any number of things that can cause a bottleneck effect here. Now, survivals themselves typically constitute a random genetic sample of the original population. And as with the founder effect, does result in a net loss of genetic variability. So let's look at what we have here. We have some sort of parental population. It's represented as beads, and they're inside a bottle. Right. And the bottleneck effect, as I sort of mentioned, tells us that when we pour this bottle, only a small amount of these beads are going to be able to flow out. and we have some limited number of surviving individuals. And then the next generation, where there is no bottleneck effect, the population comes to be. And you can see here, we've lost completely this red individual, right? So we've lost genetic variability. And that's what the bottleneck effect does. It takes your larger population and subsets it into being a smaller overall population. So in terms of genetic variability, a net negative thing. Now, let's discuss a natural case of the bottleneck effect, which we're going to look at the northern elephant seal. Its range ranges from, almost all the way up through California. Or I'm sorry, its historic, its prior range was basically in the 1800s was reduced to inhabiting the island of Guadalupe, which is this tiny orange dot off the California or off the Yucatan Peninsula here, and its current population expands all the way from halfway down the Yucatan all the way up to about 2/3 up the California coastline. Now, it had such a small range because seals and other marine mammals are tasty, they're great sources of fuel and things like that. So they were hunted to extinction, or close to extinction, in the 19th century. And as a result, because we had this pretty large bottleneck, right? It wasn't a natural bottleneck like a drought, but it was a human-induced bottleneck. It led to extreme loss of genetic variability. Now, the population, as you can see, has since recovered, but it still has very low genetic variability, even though there are tens of thousands of individuals nowadays. So, bottleneck effect again can be natural, can be human-induced, but again, it's limiting overall genetic diversity. So selection as a process does seem to favor some genotypes over others. Now, some individuals will leave behind more progeny than others, and the rate at which they do is affected by their phenotype and their behavior. Now, behavior is an interesting thing. It's something we'll cover later in the semester, but behavior is this really interesting sort of trait, but not trait, right? It's not something that's quantifiable at the genetic level, but it's certainly part of the evolutionary tract of not just humans, but also all animals. So behavior is kind of this interesting evolutionary piece as well. Now, selection comes in two flavors. Now, we've discussed both throughout this class, but we'll put actual terms to them. The first being artificial selection, which is where a human or a animal breeder selects for desired traits or characteristics within a population. And so remember, if you're selecting for a trait, there is underlying genetics to that trait. So that's artificial selection. So think about like how we have dogs, right, relative to wolves, right? That's artificial selection. Next up, we have natural selection, where the environmental conditions determine which individuals produce the most offspring. So we have this dichotomy, artificial versus natural. Now, as you probably have heard, evolution in a natural context is evolution by natural selection. So this is what Charles Darwin put forth back in the 1860s. Now, Charles Darwin had three distinct conditions that needed to be met for evolution by natural selection to occur in any given population. So, and these ones we've kind of already discussed, but let's reiterate them. So the first is variation must exist among individuals in a given population. Again, already mentioned that. Variation among individuals must result in the differences in the number of offspring surviving to the next generation. So your characteristics must affect your fecundity, right? How much offspring you can produce. And the variability must have a genetic basis. Now, there are lots of traits out there that don't have genetic bases, right? You could be big and muscular, right? And that's not a genetic trait. So that trait cannot be passed down. However, that trait can lead to differing mating conditions, right? So, but ultimately, when we're thinking about natural selection, we're thinking about how populations evolve in the wild, these three conditions need to be satisfied. Now, natural selection and evolution or process are not the same. So, natural selection and evolution are not the same. They're two sides of the same coin, but they're not the same. Natural selection is the process, while evolution is the historical record or the ultimate outcome of change through time. So again, two sides of the same coin, but they are distinct. They're not things to be interchanged with one another. And before we move on, I'll just make a note of this last point. The results of evolution that are driven by natural selection is that you have populations that become better adapted to their environment. So let's think about a hypothetical example here. We have pocket mice. They're just distinct types of mice, and we have their predator, some form of an owl. Now, we have two distinct environments here: lava rock and sand, right? And they're pretty distinct, right? Dark, light. Now, you can imagine those that are light are not favored to be able to survive on dark rock, and those that are dark are not favored to survive on light sand. So you can imagine this environment favors one type of trait over the other, right? Light here, dark here. And why is that? Well, because if you're a light mouse, you're standing out like a sore thumb, right? You're easily going to get picked off by this owl here, and if you're a dark mouse, you're easily going to get picked off by an owl here. So in this case, we have a natural selection. This selection is the avoidance of predation, and you can imagine light mice that stay here are less likely to be picked off, and vice versa. So we have selection to avoid predators. So it's kind of this nice thing, right? It can lead to selection and change over time of this population. Now, that's a physical trait. There's lots of underlying traits that you don't see, right? Your body is a really complex thing. A squirrel's body is a really complex thing. So there's lots of things that occur underneath the hood of your cells that you don't quite think about. But so one of the things that we commonly see natural selection act upon is enzymes. So the proteins inside your cells and all living things that do action, right? That catalyze chemical reactions. Now, allele frequency of enzymes actually varies with latitude. And so, for instance, allele frequencies of the enzyme lactate dehydrogenase actually in fish varies geographically. And enzymes that are formed by these alleles function differently at different temperatures. No, so lactate dehydrogenase is just involved with the breakdown of lactate, which is involved with anaerobic respiration. So, which is kind of cool. Fish that are in colder environments typically produce more lactate dehydrogenase because it's colder and they have to exert more energy to swim, whereas those in tropics produce less of it, thus leading to different allele frequencies, which I think is a kind of a cool thing. We also see this for selection, more so of an artificial selection, but again, still a type of selection in pesticide resistance as well as antimicrobial resistance. So and this is due to the widespread use of these two items. We know pesticides are sprayed all the time, and we know antimicrobials are used everywhere. So when I say antimicrobials, I'm thinking about antibiotics. Now, we do know that they've been used heavily. And so one of the things that has happened is that pesticide resistance and antimicrobial resistance have gone through the roof. So for instance, the housefly has pesticide resistance to alleles, resistant alleles for a couple genes, so PEN, KDR, and DLD/R or -R. For instance, the PEN gene decreases insecticide uptake, and KDR and DLDR genes increase the target sites for insecticides. Decrease the target sites for insecticides. So again, an artificial thing selecting for changes at the genetic level. We also see this in microbes as they're exposed to more and more antibiotics. We see resistance genes popping up in these populations. So they're becoming resistant to antibiotics, which is ultimately a dangerous thing for human health. So we see these things happening, and we see them happening relatively quickly as well. And this will just sort of show you a model, right? So this is insect cells with normal resistance alleles for that PEN gene, so uptake of the pesticide. Wild, ones that haven't been exposed to this pesticide will have lots of these receptors. Those that have the mutations have very few of these receptors. And you can imagine that this individual is far more susceptible to pesticides than this individual. So kind of a fun thing, right? It's certainly a thing that plays a role in your average everyday life, whether or not you realize it or not. And as I said, evolution is the unifying theory for all biology. Nothing in biology quite makes sense without evolution. Doesn't matter what field you're in, medicine, microbes, corn, doesn't matter. It all is tied in one way, shape, or form. Now, we can measure how well an individual is doing in the environment by this term called fitness. And we don't think of fitness in the terms of I go to the gym a lot, thus I'm fit. We think of fitness in an evolutionary context as that when an individual has a given phenotype, they will leave more surviving offspring in the next generation than individuals with an alternate phenotype. And so that's an important distinction, right? Fitness, the way we think about it, is just exercising, right? But fitness in a biological context is thinking about the survival of your offspring. I'm sure you guys have probably heard this before, but the whole purpose of life is to mate and pass down your genes to the next generation. And that's really the gist of it, from a biological concept, of course. So fitness ties directly with that idea of passing your successful genes on to the next generation and having your offspring survive. Now, it is a relative concept. The most fit phenotype is simply the one that produces, on average, the greatest number of offspring, because the more offspring you produce, the more likely there are to survive. And fitness doesn't always account for success in terms of body size or things like that. So fitness, again, is relative. Now, we can measure fitness simply by and what we typically do is assigning the most fit phenotype with a fitness value of 1. So if you have a fitness value of 1, you're the most fit individual. And so what we can do is just apply this to a hypothetical scenario. So we have a population of toads, both green and brown. Now, green toads have 4 offspring. Brown toads leave 2.5 offspring. On average, it's not you obviously can't leave half a toad dragging itself around, but again, on average. So the green fitness has to be 1. So 4 divided 4 is 1, whereas brown fitness is 2.5 divided 4, or it's 0.62. So clearly, just as a number that we have assigned, green has higher fitness than brown. But we can also see that simply by the number of offspring that are left behind. Now, fitness outside of the number of offspring has some other components that we will think about, including survival, so your capacity to survive in the environment, sexual selection, so some individuals are simply more successful at attracting mates than others, the number of offspring per mating, and traits favored for one component may be a disadvantage for others. So there are some other components to this, but again, by and large, we're thinking about successful offspring here. Now, selection itself does favor phenotypes with the greatest fitness. So phenotypes with greater fitness usually increase in frequency. So again, we're thinking about those mice. If we have brown mice trying to live on black rocks, they're not going to be the most fit for that environment. So their frequency is going to decrease in the population, while the frequency of the black mice will increase in that population. So now we can look at this as another example. So we can look at water striders. So you know if you've ever been to any body of fresh water, you've seen a water strider before. They're the cool things that skate on top of the water. They're cool looking. Now, as a note, as a generality, larger female water striders lay more eggs per day. So we have the number of eggs per day, and the the length of an adult female water strider here on the x-axis. And you can see there's a linear trend. Thus, as I increase the length of the female water striders, I will thus increase the number of eggs produced. Now, in addition, large female water striders survive for a shorter period of time. So you can see this lifespan of an adult female on the y-axis here, with the length of the adult female on the x-axis. And you can see it's not as perfectly linear as this one, but again, we see a negative linear trend between lifespan and size. So the larger you get, the smaller a chance of survival you have. And that just makes sense, right? If you're a big water strider, you're more likely to be seen by a predator and eaten. So as a result, in this population, intermediate size females produce the most offspring over the course of their entire lives and thus have the highest fitness. So we can look at this number of eggs laid during the lifetime, the length of an adult female, and you can see there's an apex here at about 14 millimeters, which is about smack dab in the middle. Again, it's a trade-off between how much eggs you're going to lay, which is, again, a measure of fitness, but also by another trait, how long you're going to live, right? So it's not always going to be the biggest females because they're not going to live the longest, but the ones that are big enough to lay a lot of eggs, but also small enough that they're not going to be picked off by predators. So we have this, this little conundrum here. Now, the reproductive strategies do play a role um, in how selection works, in particular, what we call sexual selection. Now, sexual selection, as its name implies, is selection based upon sexual behaviors. Now, males and females typically differ in how they attempt to maximize fitness. And this is not just true for, say, like fiddler crabs or, um, squirrels. It's different in the human population too, right? Humans try to attract mates differently between males and females, right? And that's what we're getting at here, right? And so females typically evaluate a male's quality and then decide whether or not to mate. Because in the vast majority of species, the females are the ones that are putting the most upfront investment. So, which is, makes sense, right? If you're going to be carrying a child for 9 months, you should be the most selective for for your mate, right? Now, pheasants also prefer. Um, um, I'm sorry, had a word. I had a word brain fart there. Peahens, sorry, not pheasants, prefer to mate with peacocks. So peacocks, they're same species, females, males, but peahens prefer to mate with peacocks with more eye spots on their tail feathers. So they're selecting their mates based upon an arbitrary characteristic in the males. I'm sure you guys have probably seen that at some point, right? So we have peahens, pheasant, I don't know why I keep saying pheasants. Peacocks, right? Lots of these eye feathers, lots of these eyes in their feathers. And we can look at the number of mates by an individual male as a function of the number of eye spots in male tail feathers. So as we increase the number of eye spots, the number of mates of those given individuals also increases. So a nice little relationship here. You could think of this for a number of things, right? That we see the same pattern with, say, the size of a fiddler crab's claw and the number of mating attempts as well. So, and there's probably ways you could quantify this for the human population too, as well. Now, as I mentioned, typically speaking, females have a higher parental investment, right? And I gave the example of, human females are carrying uh, a child for 9 months, thus they invest more in that offspring. And that's true. And that's true for the vast majority of species on this planet. And it's investment in terms of energy and as well as time. So if you're carrying a child, you have to invest enormous amount of energy to bring that child from a single cell all the way up to a fully grown baby, right? Which is why pregnant women can eat a lot, right? Because they're eating for two. Now, we also invest a pretty heavy amount of time as well. So there's all sorts of things women have to do, biologically speaking, as well as, time-wise, to raise a child from, again, a single egg to, a fully grown individual. And because females have a higher parental investment, different sexes face different selective pressures. Now, males typically increase their fitness by mating with as many females as possible, which leads to females being more limited by the number of eggs that they can produce. So a female should be choosy when they are picking a male that can provide the greatest benefit. So if you're thinking about making, if you're a natural individual, say you're a bird, you can only really mate once if you're a female. But if you're a male, you can mate dozens of times, right? So if you're a female that can only mate once, you want to choose the best male because you're going to invest a lot to raise that child. And thus, you want to make sure you have your offspring having the best chance to survive for the next generation. And you could make an analogy to human populations too, right? You want to make sure if you're going to get married and have children with someone, that you want a male that can provide for your children in the next generation, right? Someone that has a job and, blah, 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 blah. So that's what we're thinking about. However, there are some cases where there is biparental care. Now, humans are an example of this sometimes. There's not always we have mom and dad taking care of it, but there are numbers of species that do have biparental care. So both parents take care of the child in the next generation. And in some cases, when you have this biparental investment, biparental care, male investment will exceed that of females. Now, male Mormon crickets, they're just crickets. They're nothing spectacular. They actually are kind of interesting group of crickets. Now, this is going to sound gross, but it's a great example of where males provide more investment for the offspring than females do. Male Mormon crickets transfer a large protein-containing packet two females during sex, and that protein containing, when I say protein containing packet, that's just a nice way of saying a large packet of sperm. So basically a large ejaculate. So they basically give this huge amount of sperm and other, semen to the females during sex, which gives them not only fertilization, but also gives the females a large amount of sustenance to sustain their pregnancy. So in this case, where parental investment is skewed towards males, the males are the choosy sex, and the males that will simply choose the larger females to leave more offspring. So again, it's all about leaving more offspring for the next generation. So, as I said, it's all about those babies in the next generation. So we can look at this, the number of mature eggs in these crickets versus female body weight, and you'll notice that as you increase the female body weight, the number of mature eggs also increases. Thus, males will choose larger females to give their monster ejaculate to, simply because they want more offspring for the next generation, because that is how, they're not measuring fitness, but that is a way of fitness, right? Again, biology's drive is to reproduce for the next generation, right? That's the drive of biology, to produce offspring. That's really all it is. So now, sexual selection does lead for competition. And so you could think about this simply as like, if you and your best friend are interested in the same guy or girl, you might compete with one another, show off to try to woo the favor of the guy or the girl in question. And that happens naturally in the environment too. Now, we can look at what we call intrasexual selection, which is a competitive interaction between members of one sex. We can look at intersexual selection, which we just consider mate choice. We can look at secondary sexual characteristics, which are such as antlers and horns that are used to combat other males, long tail feathers or bright plumage used to persuade members of the opposite sex. We can also and commonly see what we call sexual dimorphism, and we see this to an extent between humans, but it's pretty exaggerated in many species. And sexual dimorphism simply refers to the differences between sexes. So where you have potentially, like for instance, like black widow spiders, females are significantly larger than males. Or you can think about elephant seals, which we've already talked about in this class, where the males are significantly larger than the females. So just differences between sexes. And then we also have this funky one, but called sperm competition, which selects for features that increase the probability that a male sperm will fertilize eggs. So if you took two male sperms and you dropped them together with an egg at the end of a hallway, or at the end of a tube, the one that had the higher probability of swimming to fertilize the egg, that's what we're getting at here. Kind of a weird thing to think about, but that is a thing. There are species out there where the female will mate with multiple males, and that competition between sperm becomes important. So it is what it is. Now, for intrasexual selection, you want to just note this intra versus inter. They mean two different things. Now, individuals of one sex, typically males, will compete with each other for the opportunity to mate. Now, what this does lead to, and I'm sure you've seen this maybe on a National Geographic documentary, this does lead to a few successful males being the only ones being able to engage in an inordinate amount of matings, whereas most males simply don't mate at all. So the elephant seal is a great example of this, where the beach master of the elephant seal, which controls a large section of a beach and has a bunch of females living underneath them. He's the only person that's in that territory, and I say person as in he's the only male that's in that region, and he's the only one that breeds with his females. No. You could tell they're males simply because they're significantly larger than the females. It's kind of hard to see because no one else is level with these two, but the dimorphism of the elephant seal is such that the males are oftentimes 4 to 5 times as heavy as the female. So the females might be like 500 pounds, and the males might be close to 3,000 to 4,000 pounds. They're huge. So But you can see this male is fighting another male, right? This is the dominant male. These are all his ladies, and he's fighting off this intruder of a male so that no one else can come in and mate um, with his ladies. Now, we have, in contrast to intrasexual selection, we have intersexual selection, which is active choice of a mate, which leads to direct benefits of mate choice. Now, females can benefit from choosing the male that could provide the best care, and males may provide no care, but do maintain territories that provide food and refuge for the females. Which, I think inherently makes a degree of sense. Now, there are indirect benefits of mate choice, which may lead to higher quality offspring. That may do to because of resources or better genetic material. Um, Typically speaking, they'll choose the male. Females will choose the male that is the healthiest or potentially the oldest. However, this does lead us to what we call the handicap hypothesis, which states that only genetically superior mates survive with a handicap, such as a long tail, that is, say, a hindrance in flying. And that's a hypothesis. It's a hypothesis, so it's not like a theory. It's not like evolution where it's set in concrete. So it is still something that is up for debate at this time. But just to show you that there are, this field isn't settled in stone, right? It's still, for lack of a better word, it's still an evolving field in terms of selection. So now, some courtships rely on what we call sensory exploitation, and it's kind of a cool thing. And I'll give you an example using a frog in a moment, but excuse me. Where some courtship displays appear to evolve from a predisposition in females to respond to distinct stimuli. And this sensory exploitation is simply an evolution in males of a signal that exploits these pre-existing biases. Now, this is, the example I have for you is the Tungara frog. And so males can have short bursts of sound at the end of their calls, and females of this species and other related species are particularly attracted to this call. So, this is what the frog looks like. Not the most exciting frog in the world, but you can look at the frequency of their sounds and the times at which they make those. And you can see we have this frequency. That's their sort of full call. But you can also see that there's this portion of this call that is particularly attractive to females. And so it inherently kind of like tricks them in, right? So I think maybe a good example of this from like a human population is like you have a male that has, I don't know, like pretend like the best thing for females is muscles. And so you have a male that has big muscles and that attracts all the females, right? That's what we're getting at here, right? So kind of using a trait to pull in females. So that's kind of cool. Now, we think that sensory exploitation is relatively widespread. And so, there are ways to test this, and you can read this on your own about the zebra finch, but I just figured I'd make mention of it. Now, that's variation, right? But how do we maintain variation, right? How does a population keep a large variety of variability, right? We talked about that's one of the characteristics needed for evolution, right? For natural selection to occur, large amount of genetic and physical variability. So this leads us to what we call frequency-dependent selection. Now, typically speaking, the fitness of a phenotype depends on its frequency within a given population. And there are two distinct types of this frequency-dependent selection: negative frequency-dependent selection, where rare phenotypes are favored by selection, and rare forms themselves might not be, oh, my thing. Sorry, my thing that I had here is not working. I'm not sure why. Anyways, just ignore this search image thing. Sorry, and they may be preyed upon less frequently. Now, positive frequency dependent selection, where it favors the most common form, which tends to eliminate variability, where oddballs stand out. So it'd be like the brown mice on the black lava that we discussed earlier, right? So we have two distinct ones. Being rare is better, or being common is better. It just depends. So we could look at negative frequency selection in what we call Water-Boatman Now, these are, relatively similar to those water striders that we looked at before. Now, in water boatmen, fish will eat the most common color type than they would by chance alone. So we can look at the color frequency in a population and the percentage of color type eaten by fish. And we have dark brown, medium brown, and then light brown. And you can see as we increase the frequency in the population, essentially we increase the amount of predation that is occurring. So, and we can also see positive frequency dependent selection where we have this catfish. I just love this picture because the catfish is just so like he's just so happy. He's like, oh, it's like a kid seeing a shiny red ball or something like that. But this catfish is seeing this really rare fish that's attracting him to eat this fish, where he's ignoring all these other fish. So that's positive and negative frequency selection. I just love that catfish. Now, we can also have oscillating selection, where selection favors one phenotype at one time and another phenotype at another time. And so this effect ultimately maintains genetic variation within a population. So a group of birds we'll talk about, just as a note, not next class, I should say, where we're talking about the ground finches, in particular the medium ground finches of the Galapagos Islands, where birds with big bills are favored during the drought and birds with smaller bills are favored in wet conditions. And so that's just a common thing, right? Conditions change, it favors a different phenotype, but no phenotype or genotype is favored at all times, right? It's just varies depending on the environment or depending on the time of year. So fun stuff. Now, we also have this phenomenon called heterozygote advantage. Now, heterozygotes can be favored, not always, but can be favored over homozygotes. And this works to maintain both alleles, both the dominant and recessive, in the population. Now, an example of this is actually sickle cell anemia. Now, many of you might have heard of sickle cell anemia before. It's a genetic disorder of the blood. It's mostly common in individuals with heritage from Africa. Now, sickle cell is a kind of interesting disease. It causes your blood cells to sickle. So I'll show you that picture right here. So it goes normal blood cells to sickle, right? Now, it's kind of interesting. Homozygotes for sickle cell, the allele, so it's a recessive allele, I should just make mention of that. They typically die before reproducing without medical treatment because it does limit the amount of oxygen you can take in, so much so that can cause you to be completely oxygen deprived, just as a note. So that's sickle cell. Now, as I mentioned, sickle cell is mostly found in people that have heritage from Africa. It is a recessive trait. Now, you can see the sickle cell allele in Africa, the proportion of the population, 1%, 1 to 5% here in like pinkish, 5 to 10% in that sort of intermediate color of red, and then dark red is 10 to 15%, which is cool. Now, why is this really bad disease where medical care in Africa is not the most abundant thing, just simply because of how taken advantage of this continent has been by Western countries, and how poor it's been as a result of all that terrible things Western countries have done to Africa. But why, given how bad sickle cell is, why hasn't it been eliminated? Why is heterozygote advantage a thing in sickle cell? Well, it's actually kind of cool. If you overlay the geographic distribution in these black lines here of Plasmodium falciparum, which is the parasite that causes malaria. Now, malaria is the leading cause of death in Central Africa. It infects, I think, 85 million plus people every year in Africa alone. So it's really common. But you notice that where sickle cell is found, malaria is there. Now, the cool thing is those that are heterozygous for sickle cell, so they have the dominant allele and the recessive allele, actually don't suffer anemia. However, they're less susceptible to Africa. So you have this heterozygote where they have the sickle cell allele, the bad allele, which if you had 2 of it would cause you to die prematurely, at least in a impoverished area of the world. But this allele is not gotten rid of in the population because the heterozygote has an advantage. They're essentially immune, not totally immune, but they're less susceptible to malaria. which again, I think is this really cool thing, right? Just as a note, this trait where it has two effects, giving you a disease and giving you resistance to malaria is what we call pleiotropy. So it's kind of a cool thing. But again, the heterozygote can be beneficial depending on the conditions. Now, selection doesn't just act on one trait. Selection acts on traits that can be affected by multiple genes. So we think about, when we think about classic genetics that we discussed or you've seen in a general biology course, we think of one gene, one trait, right? The color of my peas is affected by one gene, right? The shape of my peas is affected by one gene. But we do know that, oops, sorry about that. We do know that the vast majority of traits are controlled by multiple genes. Now your eye colors, multiple genes, right? Lots of genes, lots of traits are controlled by multiple genes. Now, selection acts upon not just one gene that causes a trait, but it acts upon all genes that cause a trait. And the changes in the population depend on which genotypes are favored. And so this leads us to three distinct types of selection that we'll walk through: disruptive, directional, and stabilizing selection. So we can physically see this graphically depicted for disruptive, directional, and stabilizing selection. Now, they're pretty, in terms of what they do, we're looking at the number of individuals or the frequency and say body size on the x-axis here. So how, when we see a peak like this, it just means there's a lot of individuals at this given body size. So you can think of this just like a histogram. That's really all this is a histogram. So we can think of disruptive selection where you have, these intermediate body sizes, and there's some form of selection that selects against that body size. So what it does is it weeds out those with intermediate body sizes from the population, leading to a decrease in their population size. So you can see the height of this peak dips, but what it does increases the height of the outliers, so small body size and large body size. We could see a similar trend, but again, distinct here for directional selection, where the outliers, whether it's a large body size, or you can even think about this with a small body size, where you have selection for something with a very large body size, again, weeding out smaller individuals, causing the population to shift to be net larger size. And then we have stabilizing selection, which basically selects for those in the middle. So it basically gets rid of those that are outliers, where it basically makes the curve essentially narrower, right? We're going from a fatter curve here to a much more narrow curve here. So fun stuff. So let's talk about disruptive selection. Now, it acts to eliminate, as we discussed, those intermediate types. Now, we're talking about an example here where we have different beak sizes of African black-bellied seed cracker finch. Try saying that 5 times fast. Now, available seeds in this population, only available to eat by this population, only fall into 2 categories: large or small. Now, this favors bills that are one size or the other: large bills to eat the large seeds or small bills to eat the small seeds. Now, birds with intermediate sized beaks are at a disadvantage with both seed types. They can't eat the large seeds because their beaks are big enough to crack it, but it's difficult for them to eat the small seeds because it's simply too clumsy for them to efficiently process small seeds. So we can see that, right? We have these intermediate sized birds here. Again, I love these pictures because this bird just looks so sad. Look how sad he is. And this guy's so overwhelmed. Anyways, it's just too big and too small. So disrupting selection tells me that these birds in the middle with these intermediate sized beaks simply cannot eat larger or smaller seeds. Thus, the disruptive selection pushes them out of the population. Next up, we have directional selection, again, which acts to eliminate one extreme, so small beaks or large beaks. Now, this typically occurs in nature when there is environmental change. So for example, in Drosophila, so those are the fruit flies that you've definitely seen at some point in your life. In Drosophila, flies that eliminated flies that move towards light. So those that move towards light are on one extreme of the spectrum, right? Now, what this does do, selection favors the Drosophila that don't move towards the light, thus resulting in a population that have that behavior. So we can look at this as a graph, not as pictures of surprise birds or surprise seals or anything like that. The number of generations, the average tendency to fly towards light, dark, and light. And you'll see that over time, if flying towards light is bad, you see that the tendency to fly towards light decreases through generations. So again, it's a selective force getting rid of the extreme here. And that extreme is the tendency to fly towards light. Next up is our friend stabilizing selection. And again, this acts to eliminate the extremes. Now, this makes the intermediate phenotypes more common by eliminating the extremes. And so, for instance, in humans, infants with intermediate weight at birth have the highest survival rate. So those individuals that are around 6 pounds tend to have the highest survival rate relative to very small babies and very large babies. So we can look at this as a nice graph, birth pound in weights. So you can see that the intermediate is about 6 and 7. And then the percentage of births in a population. So you notice that most babies are born around 6 and 7 pounds. You'll notice that there's a nice little bell curve here. And then you can look at the red line, which is infant mortality. You'll notice that infant mortality is high in very, very small babies. So you can think of these like premature babies. And it decreases dramatically here in the center, and then starts to come back up in the very large babies. So just kind of a nice little trade-off between being intermediate and being large. So let's talk about some experimental studies about evolution. So to study evolution, we have to rely on 2 key lines of evidence, fossils or DNA. DNA being a more new thing, but fossils are still very valuable material. Now, we can also study evolution in the laboratory setting. So for instance, we could do laboratory studies on fruit flies, have been very, very common for hundreds of years. And more recently, we've seen a lot of studies starting to take towards field experiments as well as a directed evolutionary lab. So let's talk about an example of this. So we can look at natural selection in the guppy. Pull a guppy. Yep. This is what a guppy looks like. They're nothing, spectacular. They're just little small fish. Now, we're going to talk about guppy colorations to understand guppy natural selection. Now, guppies are found in small streams in northeastern, southern South America and Trinidad. So, our neighbors to the south. And some are capable of colonizing portions of streams above waterfalls. So this leads to different dispersal methods, and it leaves some species unable to swim up the waterfall. Now, these dispersal barriers create 2 distinct environments, and the cool thing is that predators are rare above the waterfall. Now, the predator of guppies are the pike cichlids. These are really cool fish. Sorry, not cichlid, cichlid. If you haven't seen a cichlid before, take a peek at them. They're pretty cool. They are rare above the waterfall. So there are some advantages to there being no predators above the waterfall, which leads to other types of fish, including the killifish, that are rarely eating the guppies. However, it makes the lack of the cichlid predators above the waterfall, making the guppy males larger and gaudier. Now, the pike cichlid is pretty common below the waterfall, and so what it does is it makes individuals below the waterfall much more drab and reproduce earlier. So we can look at this. We have the guppy above the waterfall, really, really gaudy, awesome coloration, pretty cool looking. And then we have the guppy below the waterfall, pretty drab looking. Now, just want to point out, once this little thing goes away, they're the same species, right? Poecilla reticulata. So they're both the same species. They haven't evolved into different species, but they have very different environments, different predators, which leads to different types of selection. Gaudy guppies here, non-gaudy guppies here. Now, we can take those guppies and stick them in a laboratory session, right? A laboratory environment. Now, this is natural. Now, the waterfall is one in particular component of this. However, what if there was other factors at play here? What if up here there was just more food, which led to the guppies being more gaudy, right? There could be other factors, not just the waterfall. So we could take these guppies and pull them into the laboratory environment and remove any other potential potential confounding effects to their selection. So let's talk about this in the lab setting. Now, we can start by taking 10 large pools of guppies, and to those 10 large pools, we can add those pike cichlids to 4, killfish to 4, and 2 that are left as no controls with predators. Now, we can let those sit for 14 months and 10 guppy generations later, and it's kind of interesting. So the guppies that are found in the killfish and control pools are larger and more colorful, so they're like above the waterfall ones. And the guppies in pike cichlid pools are smaller and drab, much like it is below the waterfall. So we can confirm our field experiment that the variation between populations in nature were the result of genetic differences, not just the environment, which is pretty cool, right? Um, and so you can test what's happening in the lab in nature to see what the underlying thing is. So let's look at this. So the duration of the experiment in months, this number of spots per fish, which we define as being gaudy or not gaudy. So high number of spots is gaudy, low number of spots is not gaudy. So we have no predation in blue, low predation in red, and high predation in yellow. And you can see over time that the trend for high predation is to decrease the number of spots, i.e. less gaudiness, and as those with no or very low predators increase in gaudiness over time. So kind of a fun thing, right? The other thing I'll make mention of is that they just gathered a standard population, right? So a similar population to test this, right? They didn't just say, oh, I'm going to choose gaudy ones and then go from there. Just as a note. Now, there are many interactions um, among evolutionary forces. Now, mutations in genetic drift as processes may counter selection, which I think is an interesting concept. Now, what do I mean? Now, mutation rates in nature are rarely high enough to counter selection, but they can be under certain circumstances. And remember, selection itself is non-random, but genetic drift, i.e. random changes in allele frequency, is random. Now, drift itself may decrease an allele favored by selection, and selection typically overwhelms drift, except in a small populations. So random events are much more important in small populations relative to larger populations. Now, gene flow can also affect our friend selection, and gene flow can be constructive, where it spreads beneficial mutations to other populations. So pretend you have a really beneficial trait in North American guppies, and it gets spread to South American guppies. Remember, that's what gene flow is, right? Moving genes from one population to the next. We can also have constraining gene flow, which can impede the adaptation by continual flow of inferior alleles from other populations. So, pretend you have, again, this guppy situation where you're moving bad alleles from one population to the next. Again, counteracting um, counteracting our friend selection here. Now, let's talk about an example of this. So we're going to look at slender bent grass, and this is all it looks like. It's just a grass. It's nothing spectacular. It's just bent like that and that. That's why it's bent grass. Now, slender bent grass is found in a lot of different places, but it's found in particular at copper mines. Now, as you can imagine, copper mines, they have very high concentrations of metals in the surrounding soil. That makes sense, right? If there's a mine, there's going to be byproducts of that mine spread throughout, right? Just because humans we're bad miners. Now, the alleles of certain genes confer the capacity to grow in soils high in heavy metals. However, resistant individuals will grow slower on uncontaminated sites, which is pretty cool, right? So this trait that benefits you in polluted conditions hinders your capacity to grow in unpolluted conditions, right? I think that's pretty cool. Now, what we should expect is resistance allele to be 100% at the mine site and 0% elsewhere. However, what we do see is that the resistance allele occurs at intermediate levels in many areas that are not contaminated. And what we see is that there's gene flow between sites there is much, much higher copper tolerance than there is outside of the mine, as you'd expect, right? If you're going to grow where the copper mine is, you're going to want to have copper tolerance. Now, what we can look at is distance upwind from the mine. So when we say upwind, it means that the pollen from these grasses will not be carried this direction. So you see that the copper tolerance is low upwind of the mine site. And then we can look at copper resilience downwind of the, I want to say the iron mine, but it's the copper mine. And you can see that copper tolerance is still pretty high, right? Even as you get 160 meters away from the copper mine. Because when we say something's downwind, it means that the wind is going to be carrying the pollen, carrying the fertilized eggs, all that fun stuff away from the copper mine. So that's pretty cool, right? You can have this trait that's not beneficial outside of the copper mine site being carried, away from it, right? And it gives you a disadvantage, right? So this is an example of bad gene flow, right? Where gene flow inhibits your ability to grow and to eventually produce offspring as well. Next slide. Now, there are limits to selection. Selection is not a catch-all thing. Now, multiple phenotypes, there are typically, not typically, there are commonly multiple phenotypic effects of alleles, where the same gene affects, say, the size of a hen's comb and at a rate at which she lays eggs, right? So there is this selection for hens that lay many eggs, but have a small comb would be difficult. There's also lack of genetic variability. Now, gene pools of, say, thoroughbred horses are very, very limited. So those that win the Kentucky Derby and all of those races that force horses to run, right? The gene pool of those horses are limited, and the performance times have not improved for more than 50 years. So the horses running nowadays are not much faster than the horses running back in the 1960s, or I should say the 1970s. So, it's common, and it's common not just about, say, like purebred racehorses. It's also common in things like endangered species, right? Now, we do know that phenotypic variation may not always have a genetic basis. So there's interactions between genes. It's a phenomenon we call epistasis. There's also epigenetics, so a process by which genes are turned on and off depending on the, things that happen in your life. So there are other aspects to genetic variation that we're not going to talk about, but they do play a role here as well. So we can actually look at this phenomenon in the racehorses. So the finish time of horses in seconds. So, back in the 1880s, all the way up to modern times. Well, about modern times. This graph was probably 5 years old at this point. We could see that, modern horses were finishing the race in historic horses were finishing the race in 160 seconds. By the time the 60s and 70s rolled around, horses were finishing in about 125 seconds. And that really hasn't improved. Right? The gene pool is just too dang small, because the reality is you don't want to breed your top quality racehorse with some other horse that might have some better characteristics. You want to make sure that prize winning horse is always producing prize winning things. Now we can also look at some other traits. So we can look at the number of Omitida, I think I said that right, in fruit flies. And this, well, actually not fruit flies, just flies in general. It simply actually doesn't have a genetic basis. So you can have lots and lots of these Omitida on the right side, but on the left side. And the more you have, the better vision you have. So it, again, doesn't seem to have a genetic basis, but there is this thing that occurs as well. So this would be a trait, again, that you couldn't pass down to the next generation. So that's going to be all for today's lecture. So we'll pick up next week. We're going to talk more about Charles Darwin. We're going to talk about the origins of the species, and we're going to dive into more topics about evolution. Because again, evolution is going to be our backdrop for basically the first 4 weeks of this course. So with that, I hope you guys learned a little bit of something, and I hope you have a great day. Take care.